Thanks for making it despite another episode of Snowmageddon in Salt Lake City. We're going to focus on another aspect of SNP and indel calling today. Last time we talked about the basic challenge of distinguishing true genetic variation from sequencing error, given variability in the depth of coverage, given variability in the number of alleles that are observed for one individual. So if there's 2 alternate alleles and 9 reference alleles, how do we decide whether or not that's evidence of a heterozygote? And we talked about that mainly conceptually. And what we'll talk about a little bit today is how variant callers evaluate all those different scenarios to figure out whether or not they should predict that a variant exists or not. And it uses, most of the methods today use something called Bayes rule, which is built upon conditional probability. So we'll talk a little bit about Bayes rule. And then to reinforce that, I don't have a homework written yet, but over the next couple of days, I'll send a homework out related to the use and understanding of Bayes rule. Okay, so just, okay. Modern day technology. Okay, I'm not convinced that's going to be stable, but we'll deal with it. So just to recap, remember that what we get with our sequencing data is a random sampling of individual DNA fragments from individual chromosomes, individual strands from those chromosomes from individual cells. So we have to, and all those random fragments of DNA are put on a sequencer, and our challenge when deciding whether or not structural genetic variation exists in an individual at a particular locus in the genome is by assessing the random sample of DNA fragments that are present at that site. Okay, so we end up, we get this random sample that's in a FASTQ format, and then we end up, after doing alignment with something like BWA, we've mapped to a particular locus. We've got this random sample of individual DNA fragments from the 2 haplotypes that were inherited by this individual. I'm focusing on diploid organisms here. And in this case, I mean, if all SNPs look like this in an individual's genome, the job would be pretty easy. We've got this nice mixture of reads or short DNA fragments, samples that support the G allele and some support the A allele. And this follows our expectation that we set up with coin flipping. You know, if you do lots of coin flips, by analogy here, lots of samples of individual reads, it converges to that nice 50-50 ratio that you would expect if an individual is truly heterozygous. The challenge is sort of the corner cases when we don't have a lot of coin flips. Maybe we only have 4 sequencing reads or 8 sequencing reads. Then you get into this question of whether or not is the data supporting real genetic variation, or is this at the tails of what you might expect by random chance? Okay. All right. So now we're going to get into the math behind making that decision, essentially. And we're not going to go too deep into the math. We're just going to focus on the concept here so you understand what the tools are actually doing and perhaps why sometimes these tools make mistakes, even though there is this nice mathematical framework. Okay, so we've touched upon this, but maybe it's worth reviewing what information do we really need to decide if a variant exists. So assuming that there's sequencing errors that are made, assuming that there's PCR bias, assuming that not every site in the genome has lots and lots of reads or lots and lots of coin flips, what are the types of things that would need to be taken into account, just off the top of your head, without looking at the slide? Or looking at the slide, if you can't come up with anything. Does depth matter? Yeah, why does depth matter? What's that tell us? What's that help us decide? Yeah.
It's giving us multiple pictures of the same locus to determine if what we see is there is actually there.
Yeah, so depth is really helping us get at this signal to noise problem. Most genomics experiments when we're trying to tease out the biology are a signal to noise problem. Our signal is what we're looking for. The signal we're looking for in this case is genetic variation. The noise that confounds that signal or muddies it or makes it difficult for us is sequencing error and lack of sequencing depth. Most of the time, that's really what we're focusing on. So depth matters. What else? What about how often the sequencing technology makes mistakes? You think that matters? Why? Everyone's very awake today. Yeah?
Does that give you sort of a reference to, I guess, check your work? Let's get the expected number of errors.
Yep. Sequencing error is another source of noise. So even if you have lots of coverage, which is one source, lack of coverage is one source of noise. Even if you have lots of coverage, if the error rate is very high or is systematic, you may not be able to trust that coverage as much. Okay? What other things do we expect? If, let's say, it's a diploid organism and we're looking for heterozygotes, what do we expect if it's a true variant? If it's a true heterozygote in that individual? Yeah, that's the null, is that we would expect a 50-50, roughly a 50-50 ratio. Obviously, if it's an odd number, as we talked about last time, you can't have exactly a 50-50 ratio, but we can use that binomial distribution to calibrate what we expect. If there's 15 reads and we see 2 alternate reads and 13 reference reads, is that within our line, within the realm of expectation, or how probable would that be, assuming a binomial distribution with a probability of success of 0.5. Okay, so yeah, we talked about some of these depth of coverage, what bases are observed. So that's another thing. If we think it's a heterozygote, that individual only inherited 2 alleles, one from mom, one from dad. If there's 4 alleles present at that site, that's probably not a great indicator that it's a real genetic variant. That's probably an indicator there's something really wonky going on there. We can't trust that. It should really be a mixture of 2 different alleles. If it's a diploid individual and it's heterozygous, maybe there's going to be a couple of reads that have, errors for a different base, but you shouldn't have 1/3 of the reads saying one allele, 1/3 saying another allele, and 1/3 saying a third allele. The only caveat to that is situations like when you're sequencing a tumor genome. Maybe you have somatic mutations which sort of violate this 50-50 expectation for inherited variation. So we also need to take into account the base qualities, which are a proxy for this other source of noise, sequencing error. Composition of strand. So if it really is a heterozygote, or a real SNP in this case, within the individual, then we should see not only a mixture of 2 alleles with high base qualities, but each of those alleles should be roughly 50-50 ratio in terms of the strands that support those alleles. If the alternate allele only comes from one strand, and there's lots of sequencing coverage, like 20 reads, but they're all on the forward strand, that also suggests something funky is happening. You would expect roughly 10 on one strand, 10 on the other. Yeah?
Sorry, back to basis observed of the locus. Like, if the person was a chimera, might you see more?
Yeah, so I'm assuming that this is a diploid individual, and we're talking about inherited variation. What exactly do you mean by a chimera in this case, though?
Well, sometimes people will, people, different, fertilized eggs can, fuse.
Oh, sure.
Yeah.
Okay, yeah, that would be an example where you wouldn't expect that. Yeah.
So what you just said at the end of there should be a mix of those strands?
Yeah.
Is that because because obviously in the one strand, like you say the plus strand, that's where you always have the same mutation, right?
No.
There were variants.
Right, so it's a chromosome that has the variant, not a strand.
Right.
So you can sample that variant on either strand. So mapping qualities help us decide whether or not these reads actually belong there. So we could have this pileup of reads, everything looks sweet. We got a nice mixture of 2 alleles, they're all on different strands, they have high base qualities, but all the reads have a mapping quality 0, which is basically saying, yeah, these reads really don't belong here. So could you trust that that is a true genetic variant at that locus? Probably not. Okay. And the last thing is the expected polymorphism rate. So in human, it's something like 1 out of 700 base pairs on average, as we've talked about. So, you know, a really sophisticated variant caller could say, well, wait a second, I see like 20 SNPs, apparent SNPs in this 100 base pair section of the genome. That's like a 1 in 5 apparent polymorphism rate. If it could zoom out and sort of look at the local context of this variant, then it could say, well, maybe I don't trust any of these variants in this whole region. This region is dodgy for some reason or another, probably paralogy, as we talked about last time. Okay, so the basic point of walking through this is there's lots of expectations that we can use to separate out signal from noise. But if you think about that, each of these, if you were to write code or a mathematical model that takes into account all these different things, it gets hairy pretty quickly. I mean, you essentially have to have some probability distribution for each of these different things. How often, when it's a real variant, do I see a depth of 10, a 30, 50, et cetera? And that really comes from the data. When it's a real heterozygote, how often do I see an exact 50-50 ratio of 2 different alleles? Does it differ when it's A's and G's versus C's and T's or G's and T's? All these different things. And that's actually taking into account, oh man, my battery's gonna die here. Looks like I'm plugged in now. Okay, so that's taking into account all these different things is why SNP calling is not perfect. We don't always get the right prediction. Now, it's pretty darn good. For Illumina data in human, it's something like 99.9, 99.5% accurate, but there's billions of sites to interrogate, so even a 0.5% error rate leads to lots of false positive or false negative predictions. Right. So I have to brag about my PhD mentor and a colleague of mine now here at Utah, which is Gabor Marth. This is a clearly very early mid-1990s website that he developed to illustrate the math and the innovation behind this paper that he published in 1999. It's called PolyBayes, and it was written not for the fancy sequencing technologies we have now, but for those Sanger capillary traces that we talked about a few lectures ago. And the challenge, and I worked on this problem, sort of an advancement to PolyBayes in my first year of grad school. The problem there was in Sanger traces, if someone is really a heterozygote, you get these 2 peaks that identify the 2 different alleles in one read, because basically you're amplifying both alleles. And the signal to noise problem there is the signal is, okay, there's 2 peaks in the chromatogram that suggest a heterozygote, but the noise is that most chromatograms have low-level second peaks just because that fluorescence is a little bit noisy. So he wrote PolyBayes as a way to find high-confidence SNPs in chromatograms in diploid individuals using math identified by this guy right here, Reverend Bayes, who basically leveraged the ideas of conditional probabilities to come up with a general framework to evaluate whether or not something is likely to be real based upon what we know already. So those are called priors. If you've ever done any Bayesian work, you could probably hear priors and posteriors. Prior probabilities are sort of the expected rate of something happening, and we can use that to calibrate whether or not what we think we're seeing is real. So Gabor published this paper, and it was a big splash because it was the first paper to try that leveraged Bayesian math to really improve the accuracy of SNP calling from Sanger traces. And I don't think he gets enough credit for this. It really changed the way the detection of genetic variation was done. basically every variant caller today using modern DNA sequencing technologies uses this basic framework. So he deserves some praise there. All right, so Bayes theorem. I ripped this right from Wikipedia. You can read, hundreds of pages about Bayes theorem. An example of using Bayes theorem is given that it's sunny today, what's the probability that it's going to rain tomorrow or be sunny tomorrow? And you can use prior data. Let's say you're some weather person and you track every time it's sunny, you look the next day and ask how often does it rain. So that's our prior expectation. And then we can use that to evaluate whether or not, when the sun comes up in the morning, whether or not that's evidence, strong enough evidence to really believe that it's going to be sunny today, or there's some chance that it's actually going to rain. So you basically, the math framework is set up like this. I don't know if you've ever seen this notation, but this is saying that the probability that some random event A, could be anything, SNP, sunny or rainy, a basket is going to go in based upon a steal. Some random event A, what's the probability of some random event A given that some other random event B has happened? We know that B has happened, so what's the probability that A happens given that B has happened? And the innovation is here, you can flip this around to say, well, what's the probability of B happening given that A happened, times just the probability that A happens at all. So this is getting back to sunny, rainy expectation. How often in a year is it rainy in Salt Lake City? How often is it sunny? And those priors will differ. Salt Lake City has what, like 300 sunny days a year? Where I grew up, there were like 3. Okay, so those expectations are very different. And then, so we divide all that by the probability of A. So essentially these are the two priors. How often does A happen? How often does B happen? And then we're essentially conditioning the probability, oh my lord, the probability that B happens given A. All right, and we'll walk through more concretely how this works. So Bayesian math is used frequently in all sorts of things. So the earliest spam detection algorithms in your inbox, we're basically looking for asking how often, what's the probability that this email is spam given that a certain set of words are present in it. So I've used Bayes' rule to set my own spam filter. I have a rule in my inbox that says if the word unsubscribe is in the email, it's spam. I don't want to see it. That's burnt me a few times, but it generally works pretty well. So we can flip this around and say, well, what's the probability of seeing those words if it is spam, period, times the probability of spam. So what's the probability of spam? How could you estimate that? Yeah, you could have some poor schmo look at 1,000 different inboxes and just label emails that come in as spam or not spam and develop a prior. And that prior, the prior probability of spam depends on the person. So essentially your email client probably has to kind of figure out how many emailing lists are you on, how many conferences did you go to so that you get on all these spam mailing lists. And then I think in this context, the most important thing is what's the probability of seeing those words, period, right? So unsubscribe, I got burnt because I was thinking, well, the probability of seeing the word unsubscribe is really unique to spam. But actually, there's a lot of mailing lists sent out by the U that have unsubscribe in them, and some of them are very important to me, and I missed some critical meetings last year because I filtered those emails out because they had the word unsubscribe in them. So it's sort of, it's a signal to noise problem. Again, there is some signal in the word unsubscribe, but there's some noise in it too. So Bayes' rule, or Bayes' theorem, just one second, allows you to evaluate these prior expectations. Yeah?
Sorry, I just wanted to know if you changed that.
I changed that rule, yes. So my spam rate has increased, but so I have to hit the delete button a lot more, but the signal is still there, which is great. Okay, so in the context, oops, let's see if I can get this to work. Oh boy, guys, I don't know what's, there we go. All right. So as I mentioned before, this is a conditional probability. That is the probability of A occurring given that B has occurred. So probability of spam given words. And we can just flip that around. We'll go into more detail in a little bit. But this is what's cool about this is it's a general framework. The challenge in using Bayes' theorem or Bayes' rule in anything is figuring out what these priors are. That's the hardest work. If you can figure out what these priors are and they're very good estimates of what those priors are, then you can use this framework. A lot of times in science, though, we might have a good estimate of the frequency of one of these two things, but not a great estimate of the other. And that's where Bayes' rule can be challenging in science. But it turns out that for SNP calling, we can develop a pretty good expectation for both of these priors, okay? And therefore, we can evaluate this conditional probability using Bayes' theorem and have fairly good posterior probabilities, which is the left side of this equation. So this is a prior, that's a prior. This is the posterior probability of A happening given that B happens, okay? All right, so we're going to walk through an example of Bayes' theorem with Legos. I borrowed this from a clever website down here on the bottom that I read several years ago. I think it's kind of cool because it gives a visual representation of what we're actually doing with Bayes' theorem. So I'll try to walk through this. All right, so we've got two Legos that I've abutted here. I did this on my kitchen counter. And there's this white space, and then there's this blue space. And the white space is what, 8 by 8? So there's 64 pegs. Okay. I got to try to do the math here. So it's what? 128 are blue, which is 8 by what? 16? Yeah, 8 by 16. Okay, so the thought experiment we're going to do here is, let's say I put this up on the wall and I'm throwing darts, but these darts are special darts where they will always attach to a peg in this space. So the probability space is just the little pegs on the Lego. And I'm not a very good dart thrower, so it's a pretty random process. So what we're trying to assess is what's the probability that when I throw this dart that can land anywhere within this constrained space, that it lands on that peg or that peg or that peg, right? So if it is truly random, what's the probability that when I throw that dart and it lands on white is 64 pegs that represent white divided by the entire probability space, which is 192 pegs total between white and blue, right? So 64 divided by 192 is 0.33. So 1/3, let's just keep this baked in our heads, 1/3 of the probability space is white, 2/3 is blue, okay? So if I throw 1,000 darts, 333 of them, the expectation, 333 of them would land in white, 667 of them would land in blue, okay? So these are priors. This in Bayesian speak, this is the prior expectation, the prior probability that we land in white and we land in blue. And a critical piece here is that this entire probability space has to add up to 1. If your probability space doesn't add up to 1, something's broken with your framework, okay? Everything we have to represent the entire probability of all the different things. Okay, so in this case it does, because we've ignored the rest of the kitchen counter or the wall that I've put this thing up on, and we're only talking about this little space. So the probability of white, the prior probability of white plus the prior probability of blue is 0.33 plus 0.67, which adds up to 1, right? Okay, I think everyone's good here, right? This is now it's going to get a little bit funky, I think. At least when I first learned this stuff, I always had to just redo it and rethink through it because it gets a little, it still honestly gets a little hairy for me from time to time. So I like having these slides because I refer back to them to calibrate my thinking. So, all right, now my kid comes in and puts this black Lego on top of the blue and white. And this changes things a little bit because now when I throw a dart, there's 3 different places that dart could land. There's white space, there's blue space, and there's black space. And so this is one of those sort of foundational pegs in Legos. It's a 2x4. It's 8 pegs, right? So the probability, naively, the probability of landing on black is 0.04, right? Because it's 8 divided by 192. Anyone have a sense of what's wrong with that? What have we done if we think about it that way?
You added more than 1.
Right. We violated the rule that the entire probability space has to add up to 1. Because now we've got 0.33 plus 0.67 plus 0.04, which if I do the, it adds up to 1.042. So that's bad. And this is where conditional probability comes in. We can't treat the black pegs, as independent probabilities because it's sitting on top, it's layered. I mean, so I depicted it this way because it's intentionally layered. There's a second dimension here where you cannot land on black really unless you're already on white or blue. Okay?
That was going to be my question. Just for this, we're saying that if you land on black, you also are still landing on black.
Yeah, so, I mean, I think what I like about this analogy, I mean, it has some flaws, but I like it about the analogy is that, this block is connected. It's fused to white and black. So it establishes that direct connection. So it really is landing on black really means that underneath, you've also landed on white or blue. So there is this condition, okay? And that's, I think, the best thing about this analogy for understanding Bayes' rule and understanding conditional probabilities. Okay, so what we actually have to do is think about the probability of landing on black condition on landing on either white or blue. So this phrase, this mathematical phrase, is the same thing as saying, what's the probability of black given that we're already on a blue peg, or given that underneath there's a blue peg, okay? So that would be the probability of black given blue. Well, what we can do is sort of ignore this white part of our probability space and just focus on this blue probability space. So we already established what the probability of being in blue is. And what is that? It's 128. Yeah. Is probability of black given blue is, well, there's 128 pegs, and two of those blue pegs also have a black peg on top. Okay? So that works out to 2 over 128. And the probability of being on a black peg, given that we're on a white peg, is 6 over 64, because there's only 64 pegs. Yeah, 6 of those black ones are in white space. Does that make sense? Okay. So what's the now we can do it in the other direction though. What's the probability of being on a blue peg underneath if my dart hit a black peg? Well, I'm showing it there, but so there are 8 pegs that are black. 2 of them have blue underneath. So that's just 2 over 8. Yeah. And this is that directionality, the 2 directions of that conditional probability, the probability of being on white if you're on black, or being on blue if you're on black, or being on black if you're on blue, or black if you're on white. That reciprocality is what makes Bayes' rule work. because we can evaluate things in both directions. Okay. All right. So I mentioned this before. The probability of this is literally coming from the Legos. The probability of being on probability of being on blue given black is just the probability of being on black given blue times the probability of blue divided by the probability of black. Okay, so it is intentionally abstract. Things are just represented with A and B because it can be anything, so long as you can evaluate these important priors. Okay, and that we walked through just now how to evaluate those priors and establish those conditional probabilities with Legos. So let's plug it in. Well, what is the probability of black given white? Well, we can flip that around and say, well, what's the probability of being on a white space if the dart, given that a dart lands on some black peg, times the probability of being on the black peg period divided by the probability of being on a white peg. Okay, and that works out to 0.75. This bit times 0.04, which comes from our original priors, divided by the fraction of the probability space that is white, which is that 1/3. So that gives a posterior of 0.09 of being on a black peg, actually, given that we've landed on white underneath. Okay? And we can do, what's the probability of white given black? It's flipping the same thing, or it's using the same priors. We can plug and chug, and we end up with 0.75, which I think makes sense visually, right? So, the Lego analogy is nice because it allows us to really concretely visualize these probabilities, but in reality, a lot of things we don't have that luxury. We can't visualize them and just like count pegs. Most of the time what we're doing in science is that we have to use code or observations from doing lots of experiments to establish what those prior probabilities are. Okay, and our hope is that those prior probabilities, how often do we see a SNP in the human genome? How often do we see a mixture of A's and G's at a true heterozygous site at a ratio of 60-40, that type of thing. We hope that those prior probabilities that we measure are well calibrated. And what I mean by well calibrated is that they're generalizable, that it's not something specific to the experiments that we've done in our lab. And if they are generalizable, then the application of Bayes' theorem in this case will work really, really well. That same math would work in the lab next to you or a lab at another university, right? When they're doing SNP calling or when they're trying to estimate, to do spam detection, right? They need to do a lot of work with spam detection to make sure that they're using the right words that represent spam, so that you don't lead to some CEO using a spam detection algorithm in Apple Mail that deletes all the emails from, I don't know, some investigator or something like that. That was a very random example. What's cool about Bayes' theorem, though, is that if all that's true, it really generalizes to lots of different scenarios. So Bayes' theorem is being used all over the world in all sorts of applications, not just in genomics, but spam detection, handwriting detection, face detection. I actually had the odd experience in December of flying to England for a meeting and went to the airport in JFK, had my boarding pass printed out, and they were like, no, it's okay. And I walked right up to go on the plane, and they just had me look in this camera, and the camera took a picture of me and printed out a new boarding pass for me. So it knew who I was. So that, you can imagine behind the scenes, it's like seeing all the signal from my face, for better or worse, and then trying to evaluate what's the probability that that's Aaron or some other person. It's kind of alarming thing. Has anyone else had that yet? I think that's the future, unfortunately. Okay. The weird thing about it too is I had no choice. It was like that's the only thing you can do. It was not like, yes, I'll let you take my picture. So it's used all over the place. I've just said all this stuff. And if you're interested in this type of thing and want to learn more, there's probably a trillion websites about Bayes rule and Bayes theorem. There's some really good ones down here that talk about if you want to follow up, because the point of this is really just to give you a conceptual understanding of what's being done. All right, so let's connect it to what the lecture is actually about, which is SNP calling. So really, from a very high level, what we're trying to evaluate using Bayes' theorem, and this was established with PolyBayes from Gabor, is what is the probability that there is a SNP at this site given the data? And the data, what's the data in this case? Is it this image from IGV, or is it something else? It's the reads, yeah. It is and what, more specifically, what is in those reads that is the more granular data that this Bayes theorem is using to evaluate the probability that it is a SNP given the data. What we talked about at slide 2. Yeah. Base qualities, mapping qualities, the bases that are there, depth, how many of them there are, etc. So I think that hints at the abstraction here. So Bayes' rule is used to, we can conceive of this probability as being simplified down to what's the probability that there is a SNP given this data, but that data is complex, it's multidimensional. So the challenge is sort of modeling all the different things that could explain the probability of that data. So let's talk about that in a little bit more detail. So we can leverage Bayes' theorem to flip this probability around, and we can say, well, what's the probability of seeing that data, the exact data at this position, assuming or given that there is a SNP? And I think in this case, it's almost a little more intuitive to think about it as, well, what's the probability of seeing that data if there's not a SNP? Which is just 1 minus the probability that there is a SNP, right? Because that gets at the signal to noise problem and finding a SNP is like if there were lots of reads and lots of high quality G's and A's, well, the probability of that data given a SNP is probably pretty high. But imagine that there was only 3 sequencing reads, 2 A's, 1 G, and that G is low quality. Well, then it's going to say, well, the probability of seeing that data given that there's a real SNP is probably pretty low, or equivalent to the probability of seeing that type of data given that there's no SNP. Right? So that ratio, if the ratio of those two things is low, then the posterior probability will also be low. And it'll basically say very little evidence when I evaluate all this to believe that that's truly a SNP. Okay. So the other prior is the probability of a SNP. Well, I've probably said this statistic 50 times in the last 9 lectures or 8 lectures. What is the probability of a SNP? Yeah. Now that prior comes for human, right? What's the prior for Drosophila? Anyone know? An estimate. 1 in 300 is it's a little more than that now, but it's like 1 in 120 now. I don't know, like in bacteria, it's probably like, I don't know, 1 in 20 or something, 1 in 100. So we have to tune that prior to the organism. And actually, some of the best variant callers that are out there, you can actually set what the polymorphism rate is. And that gets baked into this equation when it's evaluating it. Okay. All right. Whoops. So, and then we talked about the probability of the data, and that's baking in all these other probabilities. Okay. The base qualities, what type of variant it is. So we know that transitions occur in the human genome and most genomes about 2 times as frequently as transversions. So, you know, if it's low quality evidence for a transversion, it's probably going to be less inclined to call that variant than if it's the similarly low quality data for a transition, because transitions occur twice as frequently. So this prior in the data will be inflated for transitions and will be deflated for transversions. Okay? We're not going to get into how you model all that stuff. I just want you to be able to sort of think through what's happening. All right, and this gets at really what's going on in PolyBayes. It's the same equation, but it's expanded out into all the things that are modeled. And so what's modeled here is what's the probability of a given sequence in allele 1 from read 1 all the way through the probability of all the reads in all the alleles observed in all that read. So it gets expanded out to all, if there's depth of 8, then this part of the equation gets expanded out to 8 terms. If there's 64 reads, it gets expanded out to 64 reads, etc. This is what we just talked about, that prior probability of the polymorphism rate. And then what it's normalized by or divided by is the probability of seeing that same mixture of alleles and base qualities given all the other possible types of SNPs or data distributions that could exist at that site. And I know that's a bit abstract, but you really have to say, well, all right, I see these reads with these base qualities. That's some signal for polymorphism, but what are all the other ways that I could see that same mixture of data and it's not a SNP? And that's what so Bayes' rule is really signal divided by noise. Signal is what's the data supporting the hypothesis that it is a SNP, and really in the bottom the equation, what's the probability of that same apparent signal just being noise. So you can think of it as a signal to noise ratio. And that ratio loosely becomes the posterior probability. Okay. So this Bayesian statistical framework has been adopted by many other SNP callers, GATK, sand tools, FreeBayes. I think you can appreciate the Bayes here. And the person who wrote it liked it because it sounds like you're saying FreeBayes for funny reasons. GATK, SAM tools, and some of the more modern ones like Platypus and other tools do the same thing. Okay, FreeBayes is another tool that is widely used. GATK and FreeBayes are sort of the two, sort of like the Ford and GM of variant calling. This was written by a postdoc. A grad student in Gabor's lab a number of years ago, and it's another paper that has been cited thousands of times now, but has never actually been published. It's been on archive forever, for reasons that are unclear to me. GATK. So I'm going to walk through GATK in a little more detail because this is the tool that's most widely used, and it's got this huge software development team that supports it. There's a nice paper describing it from back in 2010, and it really digs into some of the other probabilities that it's trying to measure that I touched upon. So if you dig this stuff, I'd recommend reading this paper. It's widely used also because they've committed a lot of effort to software documentation. A bit of writing documentation for software is really boring, but it really helps your community of users use it well. So they've done a great job with that. All right, so what I'm going to walk through quickly is just the basic process. This is a nice figure from their website, which is their recommended workflow or their best practices workflow for calling genetic variants using GATK. And they recommend some pre-processing of the data to optimize that signal-to-noise ratio so that the SNP calls that it makes are the highest quality possible, okay? So this first phase is really stuff mostly that we've talked about. So we start out with raw reads. What format is that in? FASTQ, right? So this is FASTQ. We map them to a reference genome. We talked about how that works. We do that with, they recommend doing that with BWMM. And then they mentioned this one thing that I actually didn't talk about. It's this step called marked duplicates. So this is finding duplicate molecules that arise from two different sources. What's one source of a duplicate molecule? PCR. So this was a big deal in the early days of exome and genome sequencing because there was extensive PCR required. And so when you're evaluating the evidence using Bayes' theorem for a SNP, one of the fundamental assumptions of that is that each of the data, like the sequence alignments, are independent of one another. But if there's extensive PCR duplication, those aren't independent observations. So you can imagine if you saw 50 Gs piled up at a site, if you were unaware of the duplicate problem, you'd say, well, gee, there's 50 reads, they all are high quality. Yep, that's a SNP. But if you knew that those 50 apparent reads came from one molecule, from one actual observation, you'd probably be less excited. So this marked duplicate tool goes through a BAM file and figures out whether they're duplicates. And the way it knows it's duplicates is if the start and end of the alignment of the line molecule is the same. So you have this stack up that looks like a rectangle. They have this, it's the same start and end, so it must have come from the same molecule. Now that isn't quite true if you have extremely high coverage. There is a probability that you'd have the exact same start and end site randomly, just by the shearing of DNA. But if you've got a depth of 30 genome-wide and you've got a pileup of 30 molecules that have the exact start and end, that's highly improbable given random shearing or random transposase insertion of adapters in these molecules. Okay, that's one source of duplicates. Another source is a little more cryptic, which is this cluster. Remember we talked about the amplification of a molecule on that flow cell to increase the fluorescent signal. Well, imagine you start with one molecule, one single-stranded DNA molecule, and it's clonal bridge amplified, but for some reason those molecules start to spread out just a little bit, so it looks like there's actually two different clusters, and then that digital camera recognizes them as two different clusters, but they were actually from one original clonal molecule. That will lead to what's called an optical duplicate. And so how would you detect an optical duplicate? What do we know about, yeah?
Their barcodes be the same?
That's one signal. They should have the same barcode. What else would we be able to tease out just from the information on the flow cell? Yeah.
Would the area that the molecules taking up be expanded compared to the rest?
Yes, that's true. And they would be, remember the read IDs? It gives you the name of the machine, the name of the flow cell, the lane that the molecule was observed in, the tile, and the exact XY coordinate. So those XY coordinates of those two different reads should be very close to one another, and they should be on the same lane, tile, and flow cell. So that would give you a good indicator that they're optical duplicates. Anyway, that's probably more detail than I wanted to go into, but there's this step to mark those things so that we're actually, when we're evaluating SNPs, we're actually talking about independent observations. There's this step called base recalibration, which isn't too widely done anymore, but the idea was base callers, which assign the nucleotide sequence in the FASTQ file and the base qualities associated with them. in the early days, even up to a few years ago, the base qualities were not totally correlated with the truth. So let me give a concrete example of that. Let's imagine you look at all bases that are assigned a Fred quality score of 20. If you knew the truth, what the actual base was, you would expect that all bases with a quality score of 20 would be wrong 1 out of 100 times, right? Because a Fred quality score of 20 is, log of 0.01 is -2 times -10 is 20. But what if you actually could do that experiment, the fraction of bases that had Fred quality score 20 that were wrong was actually 10 out of 100. So you're over-reporting the probability of error for bases with quality score 20 by a factor of 10. That's bad, because those probabilities are what go into, that's part of the data that the Bayes theorem is evaluating. Okay, so the way that this was done, I actually worked on an experiment doing exactly this, which was painful. You sequence a critter that you think is entirely isogenic. So there's no polymorphism in that DNA. So any apparent difference between the reference genome and the sequence that you could get like the original C57 black 6 mouse that led to the reference genome, and that thing is inbred. You get DNA from that, you align it to the reference genome that came from that same little guy, and then you know that every time you see a difference between the reference genome and the DNA is an error, not polymorphism. Then you can go through and count, well, how many reads that were aligned in this experiment had base quality 20 or 30 or 40, and then just say, well, how many of them differed from the reference genome? That's how many were wrong. That's your error rate. So base recalibration came from doing those experiments essentially, and then adjusting the curve so that the probabilities that we're reassigning the probabilities so that there is this one-to-one correlation between the predicted error rate and the observed error rate. And usually, the Bayes calling tools were overstating the error, or understating the error rate. Nowadays, the base quality scores that are assigned are pretty high quality for Illumina data. This is ongoing work with PacBio and Nanopore data because they just haven't been around for as long, and the chemistries are moving very, very quickly. All right, so at this point, we've got a BAM file that's been aligned to a reference genome. We've marked all the duplicates, so we have independent observations, and we've recalibrated all the quality scores that are in those alignments. And now we're ready for variant discovery because you've got this nice pristine scrubbed file that's ready to go where you've maximized the signal-to-noise ratio. Basically what happens is if you have one sample that you're calling variants on, you do this thing called variant calling that produces a GVCF. We're going to table that for a second, but that's a file with all the sites in that person's genome where there's apparent genetic differences using Bayes' theorem and taking all these pieces into account. Now, the dimensionality here, the stacking, is let's say we sequence the genome of everyone in this room. We would call variants in your genome, your genome, your genome, your genome, and produce, looks like 40-ish files. Then what we do is we take all those files together and we find all the sites that are polymorphic among all of us. Because I have 4 million SNPs in my genome, you have 4 million SNPs in your genome, you have 4 million roughly, but they're not always the same site. So we take the union of all those sites, and then what we do is we go through each of the sites that were found in all of our genomes, and we try to figure out what your genotype is at each of those sites. Whether you at position 1 that's found among all of us, you are homozygous reference, heterozygous, or homozygous for the second allele that was found. Does that make sense? And at the end of the day, we get a file called a VCF file, variant calling format file, that lists all the SNPs that were found among, and indels that were found among all of us. And for each of those SNPs, we'd have 40, say there's 40 of us in this room right now, we'd have 40 individual columns that list our genotype, each of the 40 genotypes at all those sites. Okay? So that is the complete set of all the genetic variants and genotypes that were found among our genomes, assuming we had the money to do whole genome sequencing and follow this process. Okay? The haplotype caller, GATK haplotype caller, is what makes this GVCF file for each of us. It calls SNPs and indels simultaneously. It does this local reassembly that I talked about last time, especially for indels, where sometimes the sequence alignments are slightly variable. It will locally reassemble all those sequences so that you get that nice pileup of reads supporting the same thing. It works on diploid and non-diploid organisms. You can tune it that way, but typically it's used just for diploid individual samples. There's lots of different tools and subtools within GATK. It's really not within the scope of this course to go through all of them. How many of you are going to be doing variant calling in your research? Okay, is it a diploid organism? Ah, okay. So that's a nice segue for me. You should not be using GATK. So GATK really does, I mean, you can fiddle with it, but it really does assume mostly diploid. But if you have an aneuploid tumor genome that you're looking for somatic mutations in, you should use something else called MUTECT, which basically uses the same framework as GATK, but makes different assumptions in the math behind the scenes about whether there could be a mixture of alleles. So PolyBayes and MUTECT are really good for tumor genomes because they don't have this assumption. Anyone else doing variant calling and any weird organisms? No? All right. Well, this lecture was probably pretty boring for you then, but so it does cool stuff in terms of figuring out what haplotypes are. So in this, let's be concrete about that. So here are 3 sites that look to be polymorphic in this very narrow, like 20 base pair window in the genome. It will figure out, it will look at the presence or the linkage of alleles that are in the same read. And that's important because we know that each read is sampling an individual strand of an individual haplotype. So you should expect to see if reads span 2 sites that are polymorphic in the individual, you should see this linkage between allele 1 at the first site and allele 1 at the second site, etc. Right? You can use that as almost like an inherited snippet of a chromosome. So it'll use that information to scrub the genotypes and the variants that it calls. And this assembly of haplotypes also allows it to clean up the alignments like we saw last time, especially for indels. So this is a site, this is the exact same site in the same individual. This is sort of the raw alignments from BWA, kind of indicating that the Smith-Waterman algorithm here sort of kind of messed things up because each of these sequences is aligned to the reference in a vacuum, agnostic to the alignment of all the other reads at this locus. What GATK will do is take all those sequences and actually compare them to one another and realize the best thing to do is to insert this big gap, which is indicative of a deletion, a heterozygous deletion in this case, because some of the reads in this diploid individual have this gap, some don't. Okay. GATK produces this big VCF file, which we'll talk about next time, and it comes from combining all the GVCF files that were found, say, from each of our genomes. And the focus of the next lecture will really be talking about the gory details of the, yes, what are we on, our fourth or fifth gory, nasty format in genomics. But we'll talk about how that works. So we're going to finish up a little bit early today. I've been working on a homework supporting this lecture and the previous lecture. I probably will send that out by email in the next few days. Most of it's going to be focused on Bayes rule and sequence alignment. I want to make sure that everyone can understand Bayes rule and actually maybe think about how to use it in their own work. Probably be a short homework, just a couple of questions, but look out for it in the next few days. Are there, since we have a few moments, anyone have questions about the lecture or homework or anything? Yeah.
So I know, like, you do work finding parts of the genome where variants are like less likely. Has that been incorporated to the like variant callers yet, where like something might more likely be an error because you don't really expect to see a variant in that portion of the genome?
That's an interesting question. To my knowledge, no one has done that. So the question is about, we know that, so we talk about the polymorphism rate being something like 1 out of 700 base pairs in human. That's very clearly an average. So a student in my lab named Jim, and actually Simone, who is working on this problem as well, there are parts of the genome that have lots of genetic variation, way more than 1 out of 700 base pairs, and there are other parts of the genome that have way less. For instance, coding sequence probably has less on average genetic variation than non-coding sequence because it encodes proteins. Those proteins are essential to the function of the cell and the organism and development, so they probably are less tolerant of changes. The question was whether or not things like GATK or PolyBayes or other variant callers take that knowledge into account. My understanding is no. But that could easily, relatively easily be put into the framework because that prior, instead of being the prior that we talked about for SNP discovery, the probability of SNP, the prior probability of a SNP, we would think is like 1 out of 700. You could give it a file that says, here's the prior probability for this window of the genome, this window of the genome, and it just looks up what that probability is and adjusts the math locally. I don't think, it's a cool idea. I don't know if anyone's doing it. You should write that. Any other questions? Okay, well, hopefully you can enjoy the snow. If any of you ski, it should be awesome tomorrow. And see you Tuesday.